Well, first of all, I'd like to thank Alyssa and Beckman Coulter to allow me to come here and talk a little bit about our work. But by way of an introduction, I wanted to just give some context for where I work, what we do, and sort of why this type of automation is valuable for what we do. So as Alyssa introduced, I'm the Associate Director at the Center for Molecular Oncologic Pathology, which is for short is CMOP. And so we have three basic core areas that we work in. So we have basic research, which encompasses histopathology, molecular pathology, multispectral imaging techniques. We have a translational development component where we work to develop preclinical assays, and we also are heavily involved in clinical trial support at Dana Farber. And then we also, our third kind of stream of research that we work in is primary tissue, and that's actually taking primary tissue from clinical patients who are enrolled at Dana Farber and other institutions that we work with, and generating mouse models for primary tumor diseases, generating cell lines, and also doing novel sort of techniques with tissue, like ex vivo tissue culturing, which is taking the tissue, primary tissue directly from a patient, cutting it into thin slices, and then actually ex vivo have this live for a very short period of time so we can do some drug analysis on it. Very preliminary, but it's directly linked to the patient as opposed to from a cell line. And so we're really at the crossroads of basic research and clinical translation. And so predominantly what we're dealing with is archival samples, so paraffin-embedded fixed tissues from patients that are actively being collected, patients who have just been diagnosed, or patients who have consented to be part of a clinical trial, or patients who have consented to be part of a research program. And so over many years, we've developed vast archives of these materials, and in many cases, this is the only tissue that's available. So obviously the downside of FFPE is that it's subject to RNA degradation. The RNA is in pretty poor condition when we get around to using it. Extracting it and actually applying it to something of value is quite tricky. Then in terms of RNA-Seq and some of the issues we've seen with the last couple of years' experience we've had, the ribosomal RNA component is very large in FFPEs. So moving towards methods where this is completely depleted actually enhances our ability to use the sequencing data that we find for meaningful biological findings. So a big part of what we do, as I said, is in the translational development arena. And so we get access to biopsied samples a lot of the time, which are very small, very low amount of tissue available. When you extract RNA, it's very small amount. So we need to think of methods where we have low RNA input, which remove the ribosomal RNA. And then if they're going to be amenable to the clinic or be of any sort of clinical translation, they need to be automated. And so the big part of this talk is going to be about the automation of the library prep, but automation of everything in general for it to be clinically relevant is sort of the arena that we're moving into. And then most of the studies that we're involved in are primarily around looking at tumor heterogeneity and looking at tumor microenvironment. So in the tissues section you can see on the left-hand side, these areas are very small within the tissue confines, so that also is limiting in terms of the amount of tissue that you can go in. So the big push here is smaller amounts of RNA input for a library, being able to automate it and do this in a very big turnaround, high throughput volume, which isn't traditionally known with this type of application. And FFP tissues are, the holy grail for clinical diagnostics. So with that, Chris is going to talk to you about the actual script-seq method.
So I would like to thank Beckman Coulter and Michaela for putting together this wonderful opportunity to look at very real applications about how we can start to begin locked information that was locked years ago because we just simply couldn't get to it, but also to pull back the curtain and talk a little bit about what actually is going on during that process to give us better insight into where that information is coming from and how we're getting it out. So for the next few minutes of this discussion, we're going to look a little bit at what actually is going on during the process. This will give you some insight into how to adapt it to your particular type of samples, but also things to think about as you start to plan your experiments and you start to plan your automation so that you don't go through areas that others have already worked through. So today we're focusing specifically on RNA sequencing, and the conceptual workflow is essentially the same regardless of the type of sample you're doing, FFPE, of course, being today's focus. And this begins with purification. The extraction of the DNA, for example, or the RNA in this case, is no small matter, as Michaela alluded to a few minutes ago, with the need to automate the entire process. But for the next few minutes, we're going to focus on the next two steps. We're going to focus on specifically the ribosomal depletion step, followed by the generation of a library that will go onto an Illumina sequencer. And then, of course, the sequencing and informatics are critical as well, but are beyond the scope of the next few minutes. The biggest challenge in this process, as many in the room will know, is how to fit all of the different steps together. When each of the different steps come from different kits, different resources, it's very difficult to get them all optimized. And so by joining the two steps in the center, the ribosomal depletion and the library generation, we've eliminated a lot of the weaknesses and areas for loss of sample and loss of diversity that come from trying to pair other kits together. So with the content that we're going to be looking at in the next few minutes, this will be applicable for two different types of kits. Scriptsy Complete, which will be the primary focus of the slides, but it'll also be applicable to the TrueSeq stranded mRNA kit. So you have two different options here. And why it's applicable to both is that each of these two kits includes the same ribosomal depletion process. The biggest challenge that we're facing right now to unlock those answers to cure cancer, to prevent cancer, to treat cancer, as well as a host of other conditions, is that many of these sample types are just not abundant. You know, patient biopsy samples, you don't want to take more than you need to. If you're sequencing single human eggs, which is a real application, this is another challenge. With FFPE, the very nature of it being degraded from the beginning limits the amount of RNA and its diversity that you can access downstream in the information that you want. And by the very nature of FFPE, the sample's degraded. It has an RNA integrity number, or a RIN number, typically somewhere between a 3 and a 6. If you're lucky, you get a little bit more intact with a higher number, but it's a very challenging sample to work with. And so the reason that the kits that we're talking about today have become so well accepted in the market and so popular is that they were the first kits that said, that's okay, you can do it. We're going to show you how to use it. As long as you're somewhere at a 3 or higher in your RIN number, we have something to help you. How this works is a two-step process. In the first step, in the ribosomal depletion step, it is a very specific process. So how it works is essentially a probe hybridization model. And we're going to show you in more detail what that actually means. But the point to remember in the next few moments is that this is very specific. And so if you have a kit designed for human, mouse, rat, you don't want to use that with plants. We have kits for all different organisms, but be careful to choose the correct option in the beginning. So this is the first troubleshooting tip. Let's make sure we have the correct set of probes. And the reason for that is that it is a probe-based design. So when you start with your total RNA solution, you have all of your different types of RNAs, mRNA with and without a poly A tail, all your ribosomals, your microRNAs. And by introducing biotin-labeled probes into that total RNA kit, those probes bind very, very specifically to just the ribosomal fraction. And so because the probes are biotinylated, simply add streptavidin beads. All of this, of course, is in the kit. Pull them down with a simple magnet, and you're left with everything else. Now, this is important to understand because this means that if you're looking at small RNAs or microRNAs or non-coding RNAs or mRNA that does not have a strong poly A tail like you would see in FFPE, it is actually still in your sample. The only thing that's been removed are the ribosomal fragments. So at this point, you can decide how to go forward in your research. So it's all well and good to say that it works, but can we show it? And so what you're looking at here is the reality that there are multiple ways to do ribosomal depletion. And we're showing removal percentage, percent remaining after depletion compared to 2 of the other methods. So QIAGEN has an antibody-based method that works so-so if you have very high quality, very intact samples. For FFPE, that's highly unlikely. For FFPE, as you can see in the third bar on the right, you actually have greater than half of your reads still as ribosomal. The depletion just is not very good. The antibody-based method has a real weakness with degraded samples. And then, of course, there's the option on the right, but it doesn't work very well. So with RiboZero, even with badly degraded samples, you still get very good removal. And why this matters is that as sequencing throughput increases, Illumina's had some very big announcements in the past 2 weeks about greater throughput and reduced cost of sequencing. We still want to drive that cost of sequencing further down. And so if half of your reads, when you're done with sequencing, are going to be something that's not biologically informative, now, ribosomal RNA is important. I'm not saying that we don't need it to function, but it's unlikely to offer the cure for cancer. There's no indication right now that that is actually going to develop the next therapeutic. And so we want to remove that up front before we pay to sequence it. And that's the entire purpose of this part of the kit. The other reason that this particular technology has been adopted so quickly is that it was the first one to get from sample to CBOT, if you're on HiSeq, or sample to sequencer, if you're on a MiSeq or a NextSeq, in less than a day. And when we say less than a day, we don't mean a 12-hour day. We mean if you start around 9 o'clock, you still have time for lunch. You can drop your samples after having been QC'd to the sequencing core by 4 o'clock and go pick up the kids from daycare. So we have designed every process to be as efficient as possible to retain sample diversity and not take any longer than necessary. These are used in a variety of different areas. What you can see here are the most common areas being used right now and widely published. For cancer research and FFPE research, we generally recommend the top left application. It's called human mouse rat gold. If you're doing clinical studies where you wish to look at the transcriptomes of both the host and the bacteria or the bacterial infection, but not the ribosomal component of either one of those, there's an epidemiology version where it will deplete the ribosomal sequences of the human, but it will also deplete it of the bacterial portion as well. And this has really increased efficiency of data across the board for clinical studies. There are some nice papers out there on sputum extractions for cystic fibrosis that we could point you to if you're interested. But what if you're not working in one of those areas? What if you're working in an animal model that is not human, mouse, rat, or you're looking at archaea or cassava? Well, we now have a new tool that became available 1 week ago that is on our website, and the URL is located there. And what we've done is take all of our probes in these kits, build them into a database, and give you the chance to search that database to determine how well that kit will work with your specific sample prior to trying it. It's called RNA Matchmaker. It's incredibly simple. You simply upload a FASTA file with your ribosomal sequences, or copy and paste them if you prefer. Run it through the database, takes less than a second, and you get a result. Either we've got a great strong match for you, we've got a partial match, or we have nothing at this time. And it will tell you exactly which one is your best option going forward. This takes the guesswork out of it and the trial and error. That way you can get to your specific answers much more quickly. There are lots of different formats. The one recommended for human FFPE would be the first line, which is the Scripsy Complete Gold Human Mouse Rat Kit. These are available in 2 options. You can get just the depletion part, which is shown in blue on the far right. It requires a little bit higher input, though. And if you prefer lower input, if you've got less in your sample, or you simply want to make sure that you have some in reserve and you're not using your entire sample in that reaction, then we have a bundled kit, which combines the ribosomal depletion step and the library prep step, which is required next. That way you don't lose any sample diversity between the two. And to give you a sense of why the library prep method has been so well adopted by our colleagues at many cancer centers is this illustration here. Another possible workflow is on the right from a different company, and as you can see, there are many, many steps to get through. Now, when you're automating this process, it's not so much of a hurdle, but every time you have a step, you risk loss of diversity, you risk loss of sample. And so the holy grail is really to get to the point where you can take your sample to sequencing with as few touch points as possible, which is why we've developed a process that is exactly that. So in these few minutes, essentially what we're trying to share with you is that we have a directional solution available that's been highly customized for human FFPE tissue as well as other types of samples that will keep that complexity so that when you get your results, you're confident that what you're looking at is truly from your sample and not from the manipulation that occurred during the processing of that sample. We do have a new format coming out soon in conjunction with the script that Beckman Coulter is going to be touching on. These are automation-friendly kits that are in 48 reactions. And what is different about these is that they have suitable volumes for automation. So we've specifically built kits that will go directly onto your platforms, and you'll actually get the full 48 reactions rather than somewhere less. Thank you.
So now we're going to go over a little bit about how we automated the ScriptSeq kit at Michaela's lab. So why do we automate kits? First of all, everybody knows good library construction is essential for quality sequencing results. And by automating this process, we're freeing up everyone to standardize their procedure, get consistency from run to run, consistency between different end users. It also frees up your time for more data analysis. You can increase your throughput while you get the same results, reduce any opportunities that may be present for cross-contamination, and protect your end users from potential hazardous materials. When we work with somebody to automate their workflow, there are some things that we do consider when we're setting up their instrument. First of all, we like to review the protocol and we start mapping out the process. We look at incubation times and temperatures, the volumes that are going to be pipetted, the different reagent types that are being used, and any start and stop points within the protocol that they'll want to be able to store their samples and walk away. We then help them decide on a platform. In Michaela's instance, she had already had the Biomek FXP from another workflow that we had automated for. But for brand new customers, we'll actually look at their throughput and what kind of devices that they want to integrate so that we can decide which platform is best for them. Anywhere from the Biomek 4000 all the way up to our larger platform, the Biomek FXP. And then for integrations, there are several things that we can integrate. For heating and cooling, we have several different Peltier options. There's an automation-friendly thermocycler that we can put on deck. There's various devices that we can put on there for shaking. And there's also the specialty things like vacuum filtration and centrifugation if that's needed. So this is the deck layout that Michaela has in her lab. This deck layout was actually configured for her because she's using FormaPure to extract from FFPE samples. She has the static Peltier in the back left corner, an orbital shaker, a shaking Peltier, a wash station, and then positions for having labware and tips. Sarah Simons is our field application specialist who worked in Michaela's lab to automate this process. And so I'm going to speak a little bit for her right now, but I can kind of say, since I do the same thing, that the first thing Sarah did was she outlined the method in our Biomek software, which is really easy to use because we have these little GUI icons that you can set up your transfer steps or move labware around the deck. We then go into doing water testings with some like reagents or just with water to kind of get an idea of how things are going to move and flow on the system. Then we either move to expired reagents or continue doing pipetting optimization with like reagents. And then we'll start doing the sample runs Usually start out with a low number of samples and then work our way up to whatever the max throughput is that the customer wants. And then we also like to apply a basic user interface to help handle the options and input variables. So when you're moving your manual process to an automated process, there are some challenges that you can possibly see. One of them is labware selection, whether or not the labware that you've used when you're doing the manual process is going to work well in the automation process. There are some labware, it's a really flexible plastic. Usually those are the less expensive plastics that you can buy, but they're also the ones that when a gripper on a robot tries to move it, it's just going to bend the labware. Shaking versus tip mixing. Shaking will cut down on time, but certain volumes and certain labware types, it might be cross-contamination possible, so you use a tip mixing. There's also light-sensitive reagents that we have to take into consideration, or any temperature-sensitive reagents and whether or not we need a heating and cooling type out. And then the pipetting optimization, the dead volumes, what we call that amount that we put into the reservoirs and reagent sources in order to make sure that we have enough to pipette out. And then there's also the pinpointing the little manual nuances that make a protocol work and then being able to translate that onto the robot. For the ScriptSeq Complete Gold Kit low input method that Sarah developed, her aim was to increase their throughput, standardize their process with less errors and cross-contamination, and streamline their workflow. She developed 3 different methods, a ScriptSeq Ribo0 method, ScriptSeq cDNA synthesis method, and a ScriptSeq library construction method. And each one of these is capable of processing up to 96 samples. So this is an example of one of those basic user interfaces that we like to put on our methods. In this instance, it was really basic. It was really only one input, the number of samples that you wish to process. And for the Ribo0 method, it takes approximately 2 hours for the robot to process 96 samples. And part of the reason that we ask for the number of samples is that we also include a reagent calculator that will pop up. And so based on your inputs into the user interface, it tells you exactly how to make up your master mixes and where to put them in the reservoirs and how much you need. The second method, the cDNA synthesis method, has a couple more options because there's some start and stop points in the protocol. So you can perform your cDNA primer annealing. You can stop there, or you can go to your cDNA synthesis, or you can run both together. Again, a reagent calculator that will pop up and tell you exactly how you make everything and where to put it based on the options that you choose within the user interface. Then the last method is the library construction method. And you can have different areas that you can check and uncheck based on which steps you want to run. So if you have a short day and you only want to get to part of the process, then you can do that. And again, the reagent calculator that shows exactly where everything is and how much you need to make of everything. And then from here, we're going to actually look at the results that Michaela has managed to get using these methods.
Okay, so at least now you have some context for the reagents and the automation steps, so that means I can just kind of maybe focus a little bit more on the science of how we apply this. But just to give you some brief introduction to where I am and what we do. So I work at the Dana Farber Cancer Institute, and obviously our mission is to offer patients the best treatment that's available while continuing to develop basic research skills and applications that help ultimately cure a lot of these disease areas. So Dana Farber has a lot of investigators. We have a lot of research grants. We're a nonprofit, obviously, and are sort of pioneering some of the new technologies that get adopted to help translate into sort of clinical diagnostics. And so our particular center, there's maybe 8 or 9 research centers that are co-localized within Dana Farber, and then there's a series of core facilities. And so we're the Center for Molecular Oncologic Pathology, as I'd mentioned. We're actually sit between Brigham and Women's Hospital and Dana Farber itself within the Division of Medical Oncology, and we support collaborative pathology-based projects primarily in 4 sort of different arenas. So we provide comprehensive pathology annotation, anything to do with clinical specimens, morphological and molecular characteristics annotation. We do a lot in the area of genetic discovery of human primary tumors with limited materials and in real time. We are always constantly looking for novel biomarkers from any sort of preclinical trials that we use with our series of mouse models. We work with different disease groups within Dana Farber to develop specific mouse models for these groups. And then a big push of our group is that we actually start to transition some of these assays that we develop in-house to actually translate them into our diagnostic labs so that they actually get uptaken into the clinic and actually get directly to patients if they're suitable and applicable. So the cohort of samples that we use for this study is in the arena of prostate cancer. Just to give you some background, there's almost 250,000 new cases of prostate cancer diagnosed last year. And of those, there's a projection of 30,000 men would die of this. It's the second leading cause of cancer death in men, and it has a higher rate of incidence and mortality in African Americans. So this is a heavy focus of research within the prostate cancer community, is trying to understand these genetic and genomic differences and anomalies based off ethnicity. Our only established risk factor right now is age. So generally, the older a man gets, the more likely he is to develop prostate cancer, and it's quite a lot of people over the age of 65 do. And then the standard practice is to do a biopsy and diagnosis, but routine PSA testing to just check the level of PSA. So even though PSA is considered a gold standard for a biomarker of prostate cancer, they've shown in the last 10 years that there's conflicting factors associated with this, and it's not always the case that if you have a high PSA level that you will actually develop prostate cancer. So a major challenge is, in clinical research, is distinguishing between indolent forms and aggressive types of prostate cancer and whether they have a tendency to reoccur. And most of the research activities that we're involved in are trying to understand this distinction more clearly. So just to give some context for, formalin-fixed paraffin-embedded tissues. So it was estimated like 2 years ago, there's probably something like 20 million archival tissue samples that are stored annually in the US alone. But because of RNA degradation during fixation and storage, it's prevented researchers from really mining them for transcriptional profiling. So, I think it's well understood and documented that DNA is a lot easier to work with, and there's a lot of work gone in that arena, but the RNA arena has sort of lagged behind. So FFPEs, it's a routine preservation method. These collections are stored for decades and they're invaluable for biomedical research because there's clinically meaningful data available on all of these patients, pre-diagnosis in some cases, so it's a very valuable, rich cohort that's just waiting to be unlocked, as the title of the presentation says. However, the biggest drawback has been the extensive RNA fragmentation and the cross-linking that has it's a limiting factor in being able to really get in and use these samples for biomarker discovery. And so just to give you an idea, in the arena of prostate cancer, sort of in generalized on the left-hand side, and then specifically around the cohorts and the groups and the collaborators we have. On the left-hand side, typically for diagnosis, they usually take 2 specimens that are collected. They take a fine needle aspirate, which is a very small amount of tissue. It's less invasive, takes a small number of cells, and it's just studied for morphology to detect whether this person has prostate cancer or not. If they do have it, they go and take a core biopsy. This is a much larger tissue punch, and they sometimes take these in multiples. It's not only one. And it's always fixed, so that's always fixed for immunohistochemistry work, for anything that they may want to look at targets on that particular specimen. And in some cases, you're able to get frozen material, although if you imagine in the hospital setting, in the clinical setting, asking how that process and protocol works to be able to go and take frozen samples and provide all the storage facilities inventoried and everything. It's a very big undertaking to ask hospitals to sort of change all of this and find budgets to do it. So we understand that even though in some cases you can ask for frozen tissue or you can get patients consented to this, it's really on an ad hoc basis, and we don't see in the future anybody really sort of making that the gold standard. So the gold standard's fixed tissue, which, you know, as we talk about, has its limitations. Prostate cancer can't be diagnosed on frozen sections, but if you have the frozen material, it's obviously in better condition, so you can do more with it. In terms of the groups that we work in, I'm a member of the TopCap team, and so this is a group of clinicians, researchers, epidemiologists globally. We have, between us, have 5 cohorts of prostate cancer that we use to do a lot of our basic research. And so these are the Physicians' Health Study from the Harvard School of Public Health, the Physicians' Health Follow-Up Study. These have been ongoing for 15, 20 years. They have enrolled many basically physicians over time, and then as this has evolved and this has grown, you know, a certain percentage of these men have developed prostate cancer. So it's an invaluable tool to study sort of from baseline level all the way through to advanced disease. And then Swedish watchful waiting cohort, and this is a group in Sweden who sort of pioneered archiving prostate tissue. They have a very large, valuable archive of these materials from healthy patients, as well as men who were diagnosed with low-grade disease, and just watching them over time to try and establish better when you should or shouldn't treat these patients. And then the Ireland Prostate Cancer Research Consortium and King's Health Partners in the UK. So it's a fairly big group of people. All in all, I'd say it's probably 150, 200 people. And we actively work with these groups on a number of different grants and another number of different projects. So getting back to the tissue, so the factors that influence RNA degradation in RNA. So this plot on the right-hand side, it just basically is talking about when you have no fixation, your strands of nucleic acid are intact, easy to extract. When you put them in formalin, depending on the fixation time, the storage conditions, the age, the humidity that these blocks are stored at, they become cross-linked. And the longer that they are in poorly housed conditions, the more likely that that cross-linking is extensive. So when you go to extract the material, you end up with very fragmented pieces of nucleic acid. And so historically, as I said, FFPE tissues are primarily been used for diagnosis or for protein marker-based IHC stains. So nobody ever thought to save them at 4 degrees. Even if you put a block, if you process it quickly and put it at 4 degrees, it will preserve the RNA and DNA far better. But obviously how these things have been collected for 50 years, and nobody at that time had the foresight to think this is possibly something we should do. So most of them have been stored in wildly different conditions, different humidities, and therefore when we go and extract material, the range of degradation that we see is not something that we can predict. It's random. And sometimes it's linked to age, but not necessarily. A lot of the cohorts we get come from different sites, so depending on how they were stored at those sites has a huge impact. Okay, so now onto the Beckman Coulter collaboration. So we actually did a phase 1 study with which we completed in 2002, which was just to start the idea of if we wanted to automate RNA-Seq library prep for frozen or FFPE tissues, what would that look like? And so the small pilot study we conducted was pretty successful. We basically used a small cohort of samples where we had tumor normal for 3 sets of patients. And then we used about a 500 nanogram input level, which at the time we were happy to use, but since then, you know, we've obviously moved down. And then we looked at doing it on a bench prep, and then how we would transfer that to automation and on the robot. And that worked out pretty well. And then so we decided then at that point, once we learned quite a lot from that, we wanted to figure out, okay, what are the more specific things we want to get at? And so this is what most of this talk is about today, is the phase 2 work, which is developing of a more streamlined process, something that's quicker, that has lower input levels, and that really we can start to now this year pilot a project to get this translated more into a clinical setting. Because clinic won't take it, even if the data looks great, if they can't do it on thousands of samples in a very short turnaround time, they're not going to be able to, we're not going to be able to meet together to do that. And so just to give you a summary of the phase 1 results, so from bench to robot. So on this panel here, as I said, we had 3 samples, so it's top, bottom are tumor benign, tumor benign, tumor benign, and then these axes, these are actually logged to the base 2 scales. And on the y-axis, you have the manual transcripts that were discovered, and on the x-axis, you have the automated process. And in all of the cases, through sequencing, you can see that There's about 25,000 transcript genes that were measured, and in most of the samples, it's around 16,000 that you can actually detect. And when you take those 16,000, you can see, based on these scatter plots, that the Pearson correlation coefficient is very, very high. So this gave us huge confidence in what we were doing, and so this was really what got us to move towards the ScriptSeq method. And so the project goals in this were to optimize ScriptSeq for FFPE samples, then to develop an automated method, and then to utilize the platform to prepare libraries. So a little bit about our clinical specimen cohort. So in this case, we wanted to be a bigger scale than what we did for the original pilot. So we have 16 tumor normal prostate cancer pairs. They were archival FFPE specimens. These tissues were sectioned and were prepared for H&E and reviewed by a pathologist to classify the prostate cancer Gleason score and to determine and annotate the tumor and benign regions on the tissues to give us the regions of interest that we wanted to go in and take core punches from. So for each sample, we took 3 core punches and then used the automated FormaPure method actually to automate and extract all of this in the one go. So we quantify our RNA and then QC for yield and quality. And the idea here was to move from where we originally started with 500 nanograms. 500 nanograms is okay if you have full whole mount blocks or you have large amount of tissue available. It doesn't really work if you're going to go into the arena of biopsies or samples that are very sparse, because even if you were able to get enough material, it probably is very hard to consent to have all of it exhausted and then have nothing left for anything else. So it's a constant balancing act. How low can you go without compromising the data you generate? But the goal here was to start with an input on the lowest level, which is 100 nanograms of total RNA. And so the way our cohort is split up, as I said, the pathologist reviews it for Gleason grade. So we had them separated into 3 bulk categories. A Gleason score of 3+3 is considered low grade. Somewhere in the middle, 3+4 and 4+3 is considered intermediary. And then anything above a 4+4 is considered advanced. And then based off ethnicity, we had, different, within each groups, we had both African American and European Americans represented. And so in all these cases, we sequenced all the samples in duplex with the appropriate adapters, and this was run over 4 separate lanes on different days. So just to give you an idea of what the path review looks like, so these purple and pinkish stains are called H&Es. This is what pathologists use regularly to diagnose any sort of disease state. And then what we have marked in, the pathologist reviews and identifies in green the benign areas where the normal tissue is present and the glands associated with that, and then in red, the tumor areas. And then within that, there's specific glands, which are tumor glands surrounded by a stroma and associated fibroblastic material. And so we go in and core in these areas, we triangulate the core to take them as close to each other as possible. You know, a big thing with prostate cancer is it's very heterogeneous, so it's very hard to really precisely know that you're taking the same amount of material each time. And then, as I said, we automated on the FormaPure method on the Biomek, and we used from the 3 core punches, you can see when you take the same amount of material roughly from each of these specimens, you actually get a very wide difference in terms of the RNA yield you get. You get anything from low hundreds up to thousands of nanograms, and this is not predictable. We've done this many, many times for other types of projects, and it's just not predictable based off age of the block or anything. We always see this very large spread. So you can see this poses a great challenge because you can't always get 3 cores from tissues. I think in most cases, 1 core is sort of the standard of what we want to go after. So you can see if we did 1 core here, some of these samples would drop out of our study. We just wouldn't have enough. And again, the same thing is shown with the RNA degradation. So these are bioanalyzer traces. So normally, I'm sure some of you are very familiar with what a bioanalyzer trace should look like. But a bioanalyzer trace should have 2 very distinct peaks representing 18S and 28S, which give you a nice RIN number. We pretty much see none of our samples having anything remotely like these. This particular cohort is a cohort that's older than 10 years. And so the best sort of example of what we see is probably on the bottom where you see 11B and 11T, where it's not fully degraded, but it's on its way. And then mostly you can see with increased RNA degradation, most of our samples lie somewhere around the middle, which is, it's basically mostly gone. I mean, we're in the RIN number area of 3, 3.5, and sometimes 2.4. I mean, sometimes it's really hard to even generate a RIN number. So these are really difficult samples. And it's nothing to do with our process of how we extract. We've done this multiple times. This is just what they look like. So Chris actually took you through the ScriptSeq workflow, but just to focus in on, as we said, we wanted to go into the 100 nanogram realm. I mean, there's 3 basic steps. One thing that we did look at in terms of the Beckman collaboration is Beckman have very nice cleanup beads that they use, which a lot of the kits don't always standardize as the method of choice for cleanup. So we actually tested Minilute columns and then 2 sets of magnetic beads that Beckman developed. We didn't bother with ethanol precipitation because we knew it just didn't work mostly. So we did actually incorporate this into our study as well. And then I guess the sort of alluring point of the ScriptSeq kit was that they actually had come up with an FFPE specific protocol, which most library preps just hadn't even factored it in yet. They hadn't really thought about it. So the guys at Illumina had thought of this, and this was really nice. And so we had shown that you really can get rid of very high amounts of ribosomal RNA and have very little left in the sample. So this was also a compelling reason for going with this kit. And so anytime we go to automate a process, because our samples are so precious, we don't have expired reagents or archival tissue that's lying around to test on. So it's very specific how we go about this. We usually pick a representative sample based off what we see for the RNA yields and RNA bioanalyzer trace. We pick something right in the middle, so where it's not the worst and it's not the best case. And then on the bench, we do a prep with this, and provided we have enough material to not only do a bench prep, but an automated prep too. And so we take it through each of the phases, and after each phase and cleanup, we actually do a trace to see what it looks like. But the most important thing here is on the bottom trace. So this is when we compared all the library preps at the cleanup for the amplified signal. And what you find is that on the left-hand side, the standard Minilute kit is the blue. This has this contaminating peak, which is basically adapter ligation. It's primer-dimer interactions. You have this peak there, and this is almost the same as having the idea of having ribosomal RNA in your sequencing data. This just becomes one of the most abundant things that's detected, and it can kind of mask a lot of what you see. And then when you use the beads, if you go in on the green and the turquoise trace and then actually go and look at the Ampere one, this peak is almost entirely removed. And this is actually very critical to what we do, because we have processed a lot of libraries and sequenced them with that contaminating peak there. And our bioinformatics people can come up with strategies to get rid of it, but you do lose information. And remember, you're already losing information by just having them be degraded. You don't want to lose any more. So this is actually a critical step in what we do. And then once we were happy with the manual prep, we ran that same sample as part of a very small number of samples on the automated system, just to make sure everything looks fine. And as you can see here, this is the same representative sample, and we generate a pretty nice library. And actually the libraries are very robust on our system. We have to dilute them to get the bioanalyzer trace, so we get a really nice libraries from this. And so once we were happy with that, we ran all the samples again in one batch. And then we look at the yield of these, the libraries. And again, it's the same sort of spread that you see from the initial extraction. However, they don't correlate. So it's not just because we have a low yield in our extraction process doesn't mean that that actually generates a really poor library, but you do get the same spread of diversity across it. So the extensive RNA degradation leads to, we get anything from 50 to 1,800 nanograms of library that actually is possible. And then once we generate our libraries, we use our core facility within Dana Farber. So we submit them for sequencing, and they do a qPCR validation on our samples. And so this is the data that they release. Well, they give us some data, and this is a plot I put together just to sort of show where we're at. So the cDNA is diluted, and they do twofold serial dilutions, and then they do technical CT measurements. And so each of these bars represents n equals 3 technical replicates. And you can see that even when you look at the tumor region relative to the benign region, you actually get differences. So even within a block where you're taking tissue from the same patient's specimen at 2 different locations, depending on how cell-rich that area is, also has an impact on whether it degrades quicker or not. So, and that's something we really don't understand. It's something that we're trying to bioinformatically understand from the sequencing data, but we don't know why that is. And then this is to give you, once we generate the libraries, to show you that the fragment size, in general, the fragment size is fairly uniform across all of these samples. So it just shows you that the kit works really well, even with the input being so diverse going in. Ultimately, what it's able to deliver is something that's more uniform that can go for sequencing. And then again, we look at the quality of the libraries, and again, you see that, you know, with increasing RNA degradation, you have per amounts of libraries that are generated. Okay, so in terms of the sequencing, these get submitted to our core. And then the instrumentation, we use a HiSeq 2400 platform they had there. We do paired end reads, 50 base pair reads. There's no point in going any higher in the base pair reads because of the fragmentation sizes. We've done some testing with 100 base pairs. It didn't really make any difference to what we were seeing. And then our core actually uses a basic pipeline of data analysis with top hat and cufflinks to give some basic stats and some basic transcript information. However, we work with our own specialized bioinformatics person who also generates her own basic metrics and also is looking at developing algorithms to better understand this specifically for FFPE. Rather than applying tools that are already available, she's actually looking to design tools that will be specific for this. This is just our sample list, and it's just really to draw attention to the fact that these are batched. They were running 4 separate runs at different times, and then we included a bunch of technical reps, so that would allow our bioinformatics people just be able to understand better what's going on and how, you know, by automating it, do you actually get better quality data. And so this is what, so this is the ribosomal depletion. So when we go and look at the ribosomal level in our samples, I mean, this may be a bit misleading, but the y-axis is actually at 0.1% ribosomal RNA present. And in all our samples, everything, so we're basically 99.9% ribosomal RNA free. We know from previous kits that we've used with these exact same samples that we were ranging between 20 and 60% ribosomal present in previous sequencing that was done. So this was, even by us, we were, really surprised that we were able to get to this level of ribosomal free. So this was really very nice results. And then when you go in and look at the unique reads, so these are mapped to the human genome 19. And then uniquely, we have, as we said, we did paired end reads, so you have in the R1 direction, it's bidirectional, and R2. Based off alignment, again, because of these samples, this varies widely anyway, a lot of the times with any sort of clinical specimen, but when you come to FFPE, it does vary greatly. And generally, we always see this in the R1 direction, we have much higher percentage of unique reads than in the R2. But across the board, it's generally fairly consistent. So we usually get around 60% reads when you combine the two. And then if you pull out a representative sample, you know, the actual percentage of what we have aligned. So on the left-hand side, we have one of our samples. On the left, it's the benign. On the right is the tumor. And then the bidirectional R1 and R2. We have percent alignment, and then we have percent unique alignment. And that relative difference is usually always maintained. And then on the right-hand side, it actually gives you the breakdown of what percentages we would see for a given sample. So unique intergenic, intronic, exonic, and ribosomal. And again, this is pretty well conserved for any given library prep method. And then if you go for the overall summary, so when our bioinformatics person compiled everything together, on average, we get about 75 million reads, plus or minus 17 million with the 50 base paired end reads. And we have 95% confidence in that data. And you can see from the box plot here on the right-hand side, the ribosomal component is almost non-existent. On the left-hand side, it's just all the unique reads that are found, and then the 3 bars in the middle are the distribution of the intergenic, intronic, and exonic regions. I had mentioned that we had included technical replicates because we wanted to see by automation, how these do. and they do pretty well. So on the left-hand side, we ran, for one of the samples, we had enough material that we could run 3 replicates of that particular tumor and not exhaust the sample. And so what you can see in the plot on the left is the XY scatter plots for the transcripts at the log to the base 2 scale, and you can see that the correlation coefficient is above 0.9. And then when we go to its corresponding benign matched pair, we were only able to do duplicates in this one, we just didn't have enough material, but again, you can see there's good correlation at 0.93. So this led us to believe that these samples, if you take them through the same process, automation of the extraction, library, and sequencing on different lanes at different days, you actually get the exact same data, which, you know, it's not always the case if you do it on the bench that you see it as nice. So in terms of normalization strategy, I mean, our bioinformatic team are working on different types and more complicated ways of normalizing, but essentially what we do, just to have a look at the transcripts and the genes, and ultimately if there's anything differentially being expressed, is that we take a housekeeping gene panel. And so these are a couple of the representative samples, and we're looking at how these housekeeping genes, there's 9 in this panel, how they look relative to the tumor normal pairs in each individual sample case. And then if we combine all of them together, that we have good correlation between them, and therefore, based off that, we can use that to just globally normalize all of our data. As I said, this is just a very crude tool to do basic analytics on the sequencing data, and our bioinformatics colleagues have far more complex models upon which they test many, many different theories. But even with this crude method, we have capabilities. Okay, another thing that we wanted to look at to just establish, how does our data look? So we have a hallmarks of prostate cancer progression. So we have a loosely defined panel of about 150 genes that we routinely look at. We look at these as targets by AHC, gene expression, many different platforms, and a combination of these can tell you a lot about disease progression. And just without mining the data, by just pulling these 150 genes out, we're able to get reasonably good separation of our normal and low-grade tumors from our high-grade tumors, which, you know, without doing much filtering of the data or anything, this is actually a very promising result. And then when you mine down further into that and you look at differential expression, if you look at twofold differential thresholds for this 150 gene panel, there's a subset of genes that pop out: EpCAM, Wnt5A, VEGF-A, and P63, TGF-beta, and keratin-15. And these are a lot of the ones that we would see traditionally, which we would see by gene expression in frozen tissues. So there's no surprise there, which is good news for us when we compare the tumor normal pairs. And then actually in 5 of the 6 gene cases, we have very good p-values to show that this is statistically significant and does actually separate the tumor and normal, the tumor and normal data. And so just to tie up a little bit here on the automation workflow, you know, obviously we get our samples in, we have to select the samples that we're interested in analyzing, so sample selection is very important, and QCing of those samples after we've done the form of pure prep. And then we take them down, and always, even before we do a big optimized run on a new reagent set, on a new set of reagents for library prep, we do a manual bench Biomek comparison, and then once we have that comparative analysis done, then we can just batch process this. So any of the methods that that Beckman have helped us develop, we now use routinely in the lab for different types of specimens. And so the follow-up on this is for this same cohort, we have frozen matched cases which were previously sequenced, and our bioinformatics team are looking at sort of the fresh frozen as a benchmark of what is absolutely available and known in these samples, using traditional pipelines and tools that have been developed, and then looking at what's lost in the FFPE samples and trying to figure out can you close the gap? There's always going to be a gap. But in most cases, you're not going to be able to have frozen material there, so maybe that's the only material that you have, and you have all these years and decades of clinical information available for these patients, so it's an important point to try and close that gap so that you get as much as you can out of them. with prostate cancer, as I had mentioned, that African Americans are far more likely to have lethal form of prostate cancer, so understanding that much better. I mean, a lot's known about SNPs and allele differences, but not really truly what separates both and how can you differentiate between who will develop lethal and who won't. And then we want to validate these signatures. So this is a small cohort. This is only 32 samples, 16 matched pairs. If we get a signature that makes sense, we'd like to validate this in a much bigger cohort of 1,000 samples that we have, so that we can really generate something that has power statistics. And so, obviously, there's a lot of people contributed to this. I've just present on everybody's behalf, but Svetlana is my collaborator, and she's our bioinformatics guru. Bina Lee is a Beckman Coulter scientist who helped with a lot of this. Ashley Rossi was the person who did all the grunt work in the lab, and Sarah Simons, as was mentioned, she helped automate the method. And then these prostate cancer specimens come from our collaborators, Matthew Friedman and Mark Pomerantz at Dana Farber, and then my boss, Max. And I can take any questions that anybody may have or may have for Mary or Chris.